Hi, class. Welcome back. It's Dr. Linder. We are still on neurology, and we're going to be talking about the neuron. So, when we talk about the neural system, we have two types of cells. One is called the neuron, and the other type of cell are called neuroglial cells. Neuroglial cells. And we have about 6 different types of neuroglial cells. The word neuroglial means nerve glue. Glial means glue. So the nerve glue cells, or the 6 neuroglial cells, are supporting cells to the neuron, and the neuron is the only cell that's electrically excitable. And what we're looking at here is a neuron, and it has different parts to it. Let's look at some of the key parts to a neuron. Um, we have dendrites. Dendra means branches, like branches of a tree. That's what these dendrites look like, branches of a tree. Then we have the cell body, which is this part right here. And the cell body is sometimes referred to as the soma. So it's either called soma or cell body, right? That word soma we saw in a previous video with somatic neural system, right? Soma, body. So this is the cell body. And then off of the cell body, we start to see the beginning of the axon. But the beginning of the axon, they call it the axon hillock. The axon hillock. It's almost like this dilated part coming off of the cell body. It's the beginning of the axon. It's called the axon hillock. Now, keep in mind that electrical signals are moving down in this direction, right? They're moving down in this direction. So action potential and nerve impulses come into the dendrites, into the cell body, and then the electrical activity moves down the axon all the way down to the end of the axon, called the axon terminal. And at the very end of the axon terminal, we have what's called an end bulb, a synaptic end bulb. And if I were to kind of take this section and blow it up, it just kind of looks like a light bulb, like that. And inside, there are these vesicles, and the vesicles contain neurotransmitters, a chemical called neurotransmitters. And this axon terminal, or the synaptic end bulb, eventually is going to connect to something, and maybe it's going to connect to muscle. So let's say this is muscle. So somehow the neurotransmitter that's released from this synaptic end bulb has to communicate with the muscle to make it contract. So we have these neurotransmitters that eventually will be released and communicate somehow with the muscle to make it contract. Okay, and we will cover that in another lecture when I cover muscles, and we'll cover a topic called neuromuscular junction, the NMJ, which will talk about all the magic that takes place here. Okay, so the main parts of the neuron are going to be the dendrites, the cell body, the axon hillock, the axon, and then at the very end, we have the axon terminal. And at the end of the terminal are these synaptic end bulbs, which contain these little neurotransmitters, these chemical messengers. Now, I also want you to see that the axon, right, the yellow nerve fibers here, are protected. It looks like there's some sort of insulation protecting it that's called the myelin sheath. Now, the myelin sheath would be equivalent to, let's say you have a toaster or a TV or even your iPhone or something that you're plugging in. You do not see, if you could just mute your mic, please.
Not 8 o'clock yet.
If you can mute your mic, please.
Oh.
Thank you. If you see that the um, axon here is insulated by this myelin sheath, the myelin sheath would be like the covering to the electrical wiring that you have, right? When you plug something in, you don't see the electrical wires. The electrical wires would be equivalent to the axon. The black or white covering to that is the myelin sheath. And the myelin is made up of cholesterol and fat. So fat and cholesterol in your diet is extremely, extremely important because it assists and affects the conduction of nerve impulses. This is why anti-lipidemic medication and anti-cholesterol medication is so damaging to the brain and spinal cord, whether it's Lipitor or Crestor or Simvastatin. These cholesterol medications limit and can affect your neurons, and they can lead to a lot of cognitive decline, like dementia and Alzheimer's. So this myelin sheath is produced by the cell called a Schwann cell. And a Schwann cell happens to be one of these neuroglial cells. It happens to be one of the neuroglial cells. And there is an autoimmune condition in which the body can attack these Schwann cells and break down the myelin sheath. And we call that MS, or multiple sclerosis. When the myelin is broken down, or you have an attack against your own Schwann cells, you now affect the conduction and the speed of the conduction, because the myelin sheath, its purpose is to speed up the conduction of the electrical currents. If you lose the protection around the wiring, the conduction goes haywire. And that's what multiple sclerosis is. Okay, so the neuron is the functional unit of the neural system. It's electrically excitable. It produces action potentials, which is the nerve conduction. We showed you the cell body. The cell body has a nucleus. If we go back, you'll see the nucleus is right here. Okay. And we talked about the cell processes, mostly the dendrites and the axons. So let's take a look at some of these lab pictures that have shown up on some quizzes and whatnot. So when we're looking at this highlighted structure, this is an axon of a motor neuron, because this is skeletal muscle. So what's highlighted is the axon of a motor neuron. When we look here, remember what I drew before, which was kind of like the synaptic end bulb that had these neurotransmitters in it. So right here, these are synaptic vesicles. These are synaptic vesicles in a presynaptic terminal. If this is the synaptic end bulb, that's presynaptic. And then the muscle that it would connect to, or the membrane here, would be postsynaptic, right? Pre is before, post is after. And these neurotransmitters will have an impact on the postsynaptic membrane. So these are synaptic vesicles which contain neurotransmitters. So this is pre, that's presynaptic. This side here is postsynaptic, and the space in between, this space right here in between, is the synaptic cleft. Would be the synaptic cleft. That would be the space. So when we look here, that yellow structure right in between, that's the synaptic cleft. Okay, we're looking at a neuron here. These are dendrites. This is the cell body, and that's the axon, and that's the axon hillock. So this blue structure is the cell body, but we don't see cell body as a choice, but we see soma. Soma means body. That's the cell body of neuron. These little circles here, these are called Nissl bodies. Let me see if I could show them to you here. Nissl bodies are these small little circles. They're involved in making little proteins and neurotransmitters. So right here in the cell body, those little circles are called Nissl bodies. Okay? It's not the axon axon's here. It's not the axon hillock is here. It's not the nucleus is right there in the center. And the soma would be that, right? So best choice. What's the highlighted structure here? Those are the branches of the tree. Those are called dendrites. In Greek, it means branches. This is the axon hillock, the highlighted section. Axon hillock, axon, dendrites, nucleus, cell body, or soma. Okay, now here we're looking at the yellow structure. Now let me go back here so we can make some sense of this. So here is the axon, and we can see the Schwann cells. This is a Schwann cell, and this is a Schwann cell, and this is a Schwann cell, and this is a Schwann cell. And right in between, there's a little line or a little gap. That little gap, which is not myelinated, is called the node of Ranvier. It's that little gap between each Schwann cell. So that's a node of Ranvier. That's a node of Ranvier. That's a node of Ranvier. Okay? So now when we go to this picture here, the yellow structure is the myelin sheath. The Schwann cells make myelin. That's the insulation. What's inside of it is the axon. Okay? So now we're looking at this blue structure is an axon, but it's myelinated because what's around it is the myelin. Node of Ranvier, those are these lines here. That's a node, that's a node, that's a node. An axon collateral, these are branches that come off. So right here is an axon collateral. It's an extension off, so we know it's not that. Okay. Let me go back here. Okay. So right here, the blue structure, that's the space between the Schwann cells. That's the node of Ranvier. The dendrites, right, these branches here, these conduct the impulses, remember, in this direction to the cell body. They're typically short, they're very, very highly branched, and they're unmyelinated, unlike the axon. The axon is branched. The axon is myelinated. So we can see, here's the axon. We can see the myelin sheath produced by the Schwann cell. Here is the axon. We can see the insulation. This is all myelin. This Schwann cell and this Schwann cell, and then there's the node of Ranvier right between it. The myelin is needed to speed up the conduction of impulses to get right to the end where those synaptic end bulbs are. And again, you'll have it attached perhaps to a skeletal muscle, and the neurotransmitter that's released will impact and affect the muscle. Here's the axon, conducts the impulse away from the cell body. We know where the axon hillock is. It's right there. There's the axon hillock, this portion here. The swollen tips at the very end. We know the swollen tips are the synaptic end bulbs, and they contain neurotransmitters. The neurotransmitter that's involved in making and being excitatory to make muscles contract are called acetylcholine. Acetylcholine. That is the excitatory neurotransmitter. That's what ACH is. So when I drew this before, and I said that's the synaptic end bulb, and it contains these synaptic vessels that have a protein in it, and this protein is a neurotransmitter. That neurotransmitter that it will release to affect the skeletal muscle, it's going to pass through the synaptic cleft. That's called acetylcholine, ACH. Okay, still on neurons. So when we classify neurons, we can classify them functionally and we can classify them structurally. When we talk about anatomy and physiology, it's structure and function. So functionally, this should sound a little bit familiar because in one of our earlier videos, we mentioned sensory and motor. We said sensory is afferent and motor is efferent. So sensory afferent neurons are going to transport sensory information from your skin and from your muscles and from your joints and all your organs and sense organs to the brain and spinal cord. Whereas the motor or efferent neurons are going to send information from the brain down the spinal cord to all of your muscles and glands. An interneuron or an association neuron is going to be the relay between the sensory and the motor, right? There has to be some sort of connection between the sensory and the motor, and that's what the interneuron or the association neuron is. So when we look here at this nail hitting your skin, ouch, right? There has to be a receptor for pain. That receptor is going to send the information through a sensory neuron. And the sensory neuron comes in, will relay that information to a motor neuron to make the muscle contract, to withdraw from the painful stimulus. The relay is the interneuron, this right here in the middle, this connection. Let's see if I can change the color of this, and we'll make it, let's say, yellow. So this connection from here to here, that's the association neuron or the interneuron, and that's 90% of the neurons in the body are association neurons or interneurons. So sensory input comes in, motor output goes out. The connection between the two is the association or the interneuron. Now, this is the spinal cord. This is a cross-section of the spinal cord. Now, what I want to point out to you, let's see if I can make this, let's make this blue. When we look at the butterfly in the middle, see the butterfly or the letter H? That's gray matter. That's gray matter. Everything around it is called white matter. Gray matter and white matter. White matter is myelinated. This is all fat and cholesterol related stuff. This is all white matter. It's all myelinated. And all the gray matter in the middle is unmyelinated. Okay. Now, I also want to make a line going across here. And this line through the center will separate posterior from anterior. So these portions back here, I'll call this blue, we'll make this blue. Those are called dorsal horns. This is a dorsal horn and this is a dorsal horn. Now, if there's a dorsal horn, guess what these are going to be called? These are in the front. So these are going to be called anterior horns. Those are called anterior horns or ventral horns. These are dorsal horns. These are ventral horns. So guess what? All the sensory information comes in and synapses in the dorsal horn, and all the motor information begins at the anterior horn. Okay? Now, structurally speaking, we have a structural classification of neurons, And there's three classifications. There's a multipolar neuron, a bipolar neuron, and a unipolar neuron. Now, I want to go back here just for 1 second, and I want to show you that in the sensory neuron, right here, notice that there's this swollen part of the sensory neuron. It's this nodule right here. The motor neuron doesn't have such a dilated nodule, okay? So this is a unique feature to a sensory neuron, is this part right here. This is called a ganglion or a ganglia. And all a ganglia is a cluster or a group of nerve cell bodies that are located in the peripheral neural system. If you have a group of nerve cell bodies clustered together in the peripheral neural system, we call that a ganglia. Okay, so this is actually called a dorsal root ganglia, a dorsal root ganglia. And it's always carrying sensory information. There is no ventral root ganglia, but there is a dorsal root ganglia. And this is one of the key characteristics of how you can differentiate what part of the spinal cord is posterior or dorsal and which is ventral, because when you look at the horns and then you look at the sensory and motor neurons, only one of them has this swollen tip to it, and it's the sensory neuron, which has the dorsal root ganglia, and that dorsal root synapses in the dorsal horn. Okay? So the reason why I bring that up is because when we look at these three classifications structurally of a neuron, we can look at a multipolar neuron, which is the one that we really just spent some time talking about, where we have several dendrites, we have the cell body, and we have one axon coming off of it, right? So if you have many dendrites with one axon, that's multipolar. And this is the most common cell type that we have. Several dendrites, one axon, that's this right here. That's a multipolar neuron. A bipolar neuron is when you have one main dendrite and one main axon. That's bipolar. These are found in the eye, the ear, and the nose. Okay, it's kind of like your special senses. Unipolar started off as unipolar. I'm sorry. The unipolar started off as a bipolar neuron. Now, imagine this cell body here pinching off and moving in this direction. If we take this and pinch it off, right, you end up with a cell body that's off to the side, and you have one process. So unipolar is just one process, and it develops from the bipolar. Now, if we were to take this and turn it sideways, you get something that looks like that. And doesn't that look like a dorsal root ganglia? Doesn't that look like this? It does. So a classical example of a unipolar neuron is the DRG, the dorsal root ganglia. Okay, so when we look at this, this is a neuron. What type of it? We have many dendrites, one axon. That's a multipolar neuron. And then we have the association neuron, right? Remember, you had motor and sensory. The association neurons are sometimes named by their shape. They could be called pyramidal cells because these are pyramidal shaped, or they're named after the anatomist or the histologist called Purkinje. Those are the association or interneurons. Good place to take our next break. When we come back, we'll get into the neuroglial cells since we already covered the neurons.